Hi, my name is Sarah, and my group mates Ali and Dylan are going to be presenting on neurotoxins and harmful algal blooms. So, what are harmful algal blooms? An algal bloom occurs when a population of microalgae accumulates rapidly in the environment. These blooms are often harmless but are considered harmful algal blooms, or HABs, when they are accompanied by natural toxin production or oxygen depletion. The nature and severity of an HAB depends on its persistence and composition. What causes them to form? There are several parameters to consider. They can be caused by natural environmental changes such as upwelling events, which bring an abundance of nutrients into the euphotic zone so the algae can use them to multiply, or they can be caused by anthropogenic influences, such as an influx of nutrient-rich agricultural runoff. Such pollution events have been known to cause severe eutrophication in marine coastal environments and have been directly linked to increases in frequency and severity of HABs in those areas. Why are neurotoxins made? Neurotoxins are produced by certain types of algae when they are present in large populations and then start to decay and degrade. The presence of such toxins in large quantities results in outbreaks of HAB-related illnesses. On the west coast of North America, the most prevalent of these events are amnesic shellfish poisoning, ASP, and paralytic shellfish poisoning, PSP. These events have also been known to cause fish, bird, mammal, and submerged vegetation kills, which translates to habitat destruction and decreased biodiversity. So, what specific neurotoxins are created? Cyanobacteria produce neurotoxins like BMAA, which likely plays a role in the development of neurodegenerative disorders such as Parkinson's. And the dinoflagellate Karenia brevis produces brevetoxin, which causes neurologic dysfunction with symptoms like paresthesias, vertigo, and reversal of hot and cold sensation. And several dinoflagellate species produce saxitoxins, which cause PSP, and then the diatom species Pseudonychia produces domoic acid, which can cause amnesic shellfish poisoning or ASP. And then on the right, there's a picture of the red tide, which is caused by aggregations of dinoflagellates in California. And those dinoflagellates produce yesotoxins, which can have similar effects to PSP. And then at night, it looks like this blue glowing color. So two of the more common algal-produced toxins along the North American West Coast are domoic acid and saxitoxin. And how do these neurotoxins enter the food web to cause poisoning? Domoic acid is taken up from the water by small finned fishes and by shellfish like mussels who rely on filter feeding. Animals who then eat contaminated fish and shellfish, such as whales, sea lions, seals, and humans, are then vulnerable to consuming domoic acid and then suffering subsequent ASP. And then saxitoxin can be consumed by mollusks who consume planktonic algae through filter feeding. And can cause potentially deadly PSP in humans and other higher consumers.
Domoic acid is a neurotoxin that is produced in the diatom genus of Pseudonychia. However, in 1959, it was first isolated in Japan from Chondria armata, a red algae. There aren't many uses for domoic acid. However, it was minimally used as an anthelmintic or as a parasitic drug that rids the host of parasitic organisms by either stunning or killing them without significantly damaging the host, which is shown in the little GIF right here. A significant effect of domoic acid on the human body is amnesic shellfish poisoning. This occurs by a buildup of domoic acid through the consumption of contaminated shellfish. Amnesic shellfish poisoning can result in permanent short-term memory loss. Such a case was seen in 1987 on Prince Edward Island in Canada, which is the first reported case of amnesic shellfish poisoning. In this case, people had died due to eating blue mussels contaminated with domoic acid. So now that we know about domoic acid, how is it made? The biosynthesis of domoic acid begins with glutamic acid and geranyl pyrophosphate building blocks and is catalyzed by DAB-A, a magnesium-dependent enzyme to yield N-geranyl L-glutamic acid, or LNGG. LNGG becomes oxidized by DAB-D 3 times at the 7-methyl of N-geranyl L-glutamic acid to produce 7-carboxy N-geranyl L-glutamic acid. shown here. Then this becomes cyclized by DABC with iron, alpha-ketoglutarate, oxygen, and L-ascorbate to become cyclized and yield isodomic acid. Then a putative isomerase occurs and domoic acid is the final product.
Now let's take a look at the biological mechanism of the excitatory neurotoxin domoic acid. To do that, we're going to have to answer a few questions first. What is an excitatory neurotoxin? What parts of the brain does domoic acid affect the most? How does it do damage to that portion of the brain? What symptoms come as a result of this damage? And what can we do to treat it? First up, what is an excitatory neurotoxin? To answer that, we need a little info on neurotransmitters. Neurotransmitters are the chemicals that allow neurons to send signals to each other. They are sent from one synaptic terminal through the synaptic cleft to activate receptors on the other terminal. An example of one such receptor is the AMPA receptor shown here. Excitatory neurotoxins are chemicals that interrupt the normal signaling by binding to receptor sites, like glutamate to this AMPA receptor. Depicted here is the NMDA receptor that regulates calcium concentration in the postsynaptic cleft. Ka receptor, AMPA receptor, and NMDA receptor are all ionotropic glutamate receptors, and they are targets of domoic acid intoxication. Okay, so where does damage from domoic acid occur? Well, it largely occurs in the pyramidal neurons located in the hippocampus, as well as neurons in the retina. Here we see the cross-section of a rat hippocampus showing bloated damaged cells in the CA4 and CA3 regions. So how does domoic acid cause this damage? Well, domoic acid binds tightly to the ionotropic glutamate receptors, causing an influx of calcium ions across the postsynaptic terminal and endogenous glutamate buildup in the synaptic cleft. The calcium ions flood the postsynaptic terminal, and the free glutamate activates enzymes including nitric oxide synthase, protein kinase, and phospholipase A2. The calcium influx and free glutamate cause a chemical cascade that leads to radical oxygen species, lipid peroxidation, and DNA damage, as well as causing osmotic damage to the cells, or what's known as bloating. To compound these effects, domoic acid also binds to the astrocytes responsible for regulating glutamate, so the cascade goes uninhibited. All these changes to the chemical environment interrupt mitochondria, leading to a depletion of energy and ultimately apoptosis of the cells. The results of these neuronal cell deaths in the hippocampus and other parts of the body can be severe. Symptoms include nausea, vomiting, seizures, confusion, dizziness, permanent acute memory loss, and even death. Most severe symptoms may not happen in all cases of domoic acid intoxication, but if the person does experience severe symptoms, there isn't much doctors can do besides give life support while the body works to eliminate the toxins. Here are our references, and we hope you enjoyed the video.